This is Unit 8, Part 1 of Unit 8, on the system that is involved in intake and processing and absorption of both solid and liquid. The digestive system, an overview is that you have a tract, a GI tract or gastrointestinal tract, also known as the alimentary canal, and it's involved in digestion and absorption of food, and it includes all of the following of these organs that are listed here, one after another. It's essentially a tube. It's a space outside of your body. Some students have difficulty understanding that it's not inside their body. It's not in the body tissues. That's what I mean by not inside your body. So it is an open space continuous with the outside world. There are also accessory digestive organs, which include the teeth, tongue, gallbladder, salivary glands, liver, and pancreas, all accessory organs that assist the function of the alimentary, of the GI tract. Here's an overview diagram showing the different parts, and I would say pretty much almost everything is important. If you go from top to bottom, which I think we will, we will go from top to bottom when we talk about the structure. All of these, this is in bracket, it's not part of the, it is an organ that you need to know, but it's not part of the digestive system, the spleen. Large intestine includes all of these sections, all of which you should know. So everything except the one thing on this graph that's not part of the digestive system. That doesn't mean you don't have to know it, but it's not part of the digestive system, the spleen. So all of these organs, going from top to bottom, we'll discuss them in the next few slides. The digestive process itself is essentially, it's a disassembly of polymers. The polymers, of course, are the ones you eat in food. They come from living things, so they're made of the four main types of molecules that all living things are made of: carbohydrates, proteins, nucleic acids, and lipids. And as they get digested, as the molecules are digested, broken apart by hydrolytic enzymes and acid and mechanical cutting and tearing of it, then they become available for the body to absorb. There are overall 6 essential activities that make up the digestive process. They include ingestion, propulsion, mechanical digestion, chemical digestion, absorption, and defecation. And these are summarized here on this diagram, which you could look through on your own. So in terms of activity of the gastrointestinal tract, that includes ingestion of food, taking it in, propulsion, swallowing and moving it along, and food material is moved along through the digestive tract by the action of peristalsis. Peristalsis is a wave of contraction, relaxation of the muscles in the organic wall, so you get this continuous wave of contraction that's moving forward, and by forward I mean in the direction of from the mouth towards the anus. So peristalsis is occurring. It's a reflex reaction that occurs in the gastrointestinal tract that helps move material along. During processing of food, there is mechanical digestion, and that occurs by chewing and mixing and churning of the food. Okay, so this is a diagram that shows what peristalsis is and what segmentation is. These are two movements that you get within the GI tract. Peristalsis is a wave of contraction that moves along the from the beginning towards the end of the gastrointestinal or GI tract and helps move material along. So you're getting waves of contraction. This contracts, then it relaxes, and the next part contracts, and that relaxes, and the next part contracts. So it's constantly pushing along. At the same time, I moved ahead too quickly. At the same time, you have also this, besides peristalsis, you also have segmentation, where different segments are constantly right beside each other, contracting and relaxing, and that's to more mix the material up. Other activities of the GI tract include chemical digestion, absorption, and defecation. Chemical digestion is, in fact, the catabolic breakdown of the polymers that make up food. The four main polymers that living things are made of are broken down by hydrolytic enzymes, and they're broken into their individual building blocks. So proteins broken into individual amino acids, carbohydrates into monosaccharides, lipids into free fatty acids, and glycerol and nucleic acids down to nucleotides. The absorption occurs mostly in the small intestine, and mostly in the first portion of the small intestine, which is called the duodenum. And the molecules that are produced by mechanical and chemical digestion now are small enough to get absorbed across into the body tissues, and they go into the blood or the lymph, depending on which type they are. I'll explain that later. We'll go through all of these much later in more detail, as we go along in more detail. Defecation is elimination of anything that's indigestible, solid waste. So, the GI tract is an external environment, external to your body tissues, and it's used for the digestive process. You know, there's a lot of things going on in there that if your tissues were exposed to them, your tissues would get digested, and you don't want that. So it's outside of your tissues in this space that's continuous with the outside world. It is regulated, as all physiological systems are regulated. Digestion is carefully regulated. The regulation involves mechanical and chemical stimuli and the response to those. There are stretch receptors. Well, let me see. The response, the stimuli include stretch, which can be picked up by stretch receptors. There's also changes in osmolarity when different materials are ingested, changes in pH due to presence of material, and that's what's meant by substrate. Any material within the space of the GI tract will cause a change in pH. And there are end products of digestion which will also result in a regulation of the whole process. You know, changes in the environment in the body, in different parts, are constantly detected because you are constantly monitoring the body is constantly monitoring the body systems. This is how the nervous system works, essentially. There is constant monitoring by sensory tissues, and the different sensory tissues respond to different stimuli. They're specialized. And we're talking about stimuli from outside or from within the body. So an example of an outside stimulus would be light shining into the eyes, and the eyes have lots of light-sensitive receptors that respond to different colors or intensities of light, and send signals back into the central nervous system, into the brain. So that's what's happening in your body, throughout your body, for different systems. Sensory information is constantly being detected and sent back into the central nervous system, where it gets processed, and then signals are sent out to excitable tissue like muscles or glands, so that they can respond appropriately to that stimulating initial signal. So you have sensory information going into the central nervous system, where it gets processed, and then signals get sent out to effector organs that effect some kind of change. If the signal goes in and it has to be processed, anything more than the most simple type of processing, then it'll occur up in the brain. The signal goes up into the brain and is processed. However, very, very simple responses initiated by stimuli that are picked up that trigger very quick responses and don't need much processing at all, that's what's called a reflex. So you get this reflex arc. Sensory information comes in, it goes into the central nervous system, but it'll just go into the spinal cord, and there will trigger a response, and the response gets sent out to muscle tissue or gland tissue. And that's the kind of thing that does happen in the GI tract, where sensory information coming in, different kinds of stimuli on the basis of different changes will send signals into the spinal cord and trigger response signals to effector organs, and will activate or sometimes inhibit digestive glands. There are really, in the gut, there are also some very quick reflexes that are automatic, very close by, that actually don't need any nerve tissue. You know, just stretching the smooth muscle of the wall, in the wall of the gastrointestinal tract will trigger contraction, and that helps move the contents along. So, as I said earlier, you have some built-in controls in the GI tract that can initiate short reflex systems. A plexus is just a network. So you have nerve networks or nerve plexuses right near the GI tract that can initiate very short reflexes involving just the local tissue. Sometimes, and this is slang really, it's called the gut brain. The gut does have some release of neurotransmitters, signals that are released that can trigger responses, localized responses within the gut. And of course there are extrinsic controls, longer reflexes that arise outside of the GI tract. They arise within, but they will go, the reflex arc involves going out of the GI tract and then to the central nervous system and sending signals back to the activate to the gut. So localized intrinsic controls and extrinsic controls that are triggered in the gut and go to the outside, come back in, sending signals back in. So, and this is a very, very generalized overview of what the nervous control in the GI tract is. So you do have longer kind of reflexes, and that can be triggered, for example, quite remotely from your gut, just by the sight or smell or thought of food. That can stimulate the nervous system to send out impulses to the localized networks of nerves that will trigger the effector tissue, the smooth muscle or glands in the gut to respond. So that's a kind of localized, that's a external stimulus, and then signals sent down to the gut. There are also sensory signals that involve these longer extrinsic kind of reflex arcs, where the sensory tissue, remember it's always sensory tissue, central nervous system processing, and then signal going out to the effector tissue to muscle or gland. So here we have signals picked up within the gut by chemoreceptors, chemical receptors, osmoreceptors. They respond to the tonicity or osmolarity of the solution in the space of the gut, which can change when food is present. Mechanoreceptors respond to stretching of the wall, which occurs with food moving through the wall, and that can send sensory input and information to the central nervous system. So these are outgoing impulses, outgoing sensory nerve impulses, afferent impulses going towards the central nervous system, and then triggering process, and that'll be processed in the central nervous system and then sent down to the local tissue, triggering activity in the excitable effector tissue. Remember, again, I'm always referring to very simple kind of system of incoming information processing and outgoing effectors, outgoing signals to effector organs. So you have sensory input, right? Signals are picked up by specialized sensory tissue, and some examples of that kind of specialized sensory information. Here you see this is coming from outside of the body. This is images picked up by light receptors in the eye. This is picked up, smell is picked up by receptors in the nasal lining of the internal nasal cavity. Taste is picked up by the sensory tissue in the tongue. Thought of food is actually triggered within the central nervous system. It's the one thing that there is no information coming from outside. It's inside of the central nervous system. But essentially you have sensory information. In this example here, it's from outside the body. In this example here, chemical, osmotic, or mechanical sensory information is from inside the body. But it's all sensory information that are sending signals to the central nervous system, triggering, always they get processed by the central nervous system, and they can trigger outgoing signals to effector organs. Okay, and the effector organs are always some kind of muscle or gland. The processing that occurs, there are a number of different kind of outgoing impulses, nerve impulses that are outgoing. The outgoing impulses, efferent refers to outgoing, and this is relative to the central nervous system. Afferent are incoming to the central nervous system. So these actually here are afferent signals going in. These are afferent sensory signals here. These are afferent sensory signals going in, and these are efferent signals going out. Efferent impulses, nerve impulses, or nerve signals going out, and the end result is always triggering, if required, triggering of effector organs, either muscle or gland. The difference between long reflexes and short reflexes is that long reflexes actually go up into the central nervous system, where short reflexes, they're processed locally by these nerve networks in the wall or just outside the wall of the gut. Here the stimulus can occur in the space because of changes in the space of the gut, triggering the information, the sensory information goes into the local nerve network and is enough to trigger a short reflex, and the response again is always, all responses are either muscle or gland tissue. Muscle contracts, glands secrete. Okay, so sensory input, let me just finish this one thing that I had here. Sensory input into the central nervous system, which is the processing area. Information is processed, and then efferent output to effectors and effector tissue. Effector. So difficult writing with this. Effectors. And effector tissue is always either muscle or gland tissue. Okay, In the space of the abdominal cavity, as you already know from what we've talked about in the lab, inside the peritoneal cavity you have serous membranes. The serous membranes in the peritoneal cavity are the peritoneal membranes. You have a visceral peritoneum and a parietal peritoneum. The visceral peritoneum covers the organs inside the gastrointestinal tract or the abdominal tract, and the parietal membrane or parietal peritoneum lines the cavity, the wall cavity, the abdominal cavity. In the space between the two membranes is serous fluid, so that things slide past each other. They lubricate not the space of the digestive organs, but the outer surface of the digestive organs, so that everything is nice and slippery and nothing is sticking to each other. They slide across each other. This shows you the parietal peritoneum on the wall and the visceral peritoneum on the surface of the organs. And the fact that some organs are actually hanging off of a curtain kind of mesentery. And you've seen the mesentery in the for the gut. It's that clear membrane that the loops of the intestinal tract, and it's most easily seen with the small intestine. So you can gently lift up a loop of small intestine and see that clear membrane. And embedded in the clear membrane, of course, are blood vessels sending arterial blood towards the intestine and returning venous return coming away from the intestine. The mesentery is a double layer, peritoneal membrane. It, as I said, has blood vessels in it. It also has nerves running in it, and these supply vascular and nerve supply to the viscera. The viscera are the internal organs, right, covered by the visceral peritoneum, or by the visceral peritoneal membrane. And they also not only allow for those suspension of those blood vessels nicely in the nerves, but they hold the digestive organs in place. Sometimes fat gets stored in the mesentery. It can be actually quite a bit of fat. There's a word for that fat. I can give it to you. I wouldn't hold you responsible for it. But fat that is suspended in the mesentery is called omentum, and can be quite significant amounts of it. Omentum, just for your own information. Some organs are outside of the peritoneal cavity. They are, if they're behind, more dorsal, they're retroperitoneal. Some are other organs are within the peritoneal cavity, and they are intraperitoneal, surrounded by the peritoneal membrane of the visceral peritoneum. And this is what happens during development. Initially, these organs sometimes are inside the space, but during development they become retroperitoneal. I wouldn't worry about this. We don't do much development. There is blood supply, significant blood supply to the organs within the peritoneal cavity. I did tell you that the spleen is not a digestive organ, but it is within the peritoneal cavity or abdominal cavity, and therefore is talked about. There are some major arteries feeding towards the spleen, the liver, and the stomach, and they are called the splenic, the hepatic, and the gastric, left gastric artery. The mesentery has also, arterial blood supply. There is also, and this is quite important, a hepatic portal circulation. Blood, venous blood that has picked up digested nutrients by, molecules that have been digested, broken down into their building blocks, get absorbed across the wall of the intestine in the small intestine and get absorbed into the blood, and that venous blood returns always back first to the liver, where it gets processed before it goes onward back to the heart. And that is the hepatic portal circulation, which I'll mention several times later in the course in more detail. There is a generalized overall pattern to the tissues of the alimentary canal. So the study of tissue is histology, and we will go through the different layers in general terms that are common, the common features going from the esophagus down to the anal canal. There are 4 layers, also known as tunics. So there are 4 main layers within the gastrointestinal or alimentary canal. They include the going from the space inside, right beside that space, there's mucosa. Further into the body tissues is the submucosa, then the muscularis externa, and then the serosa. Outside of that, all of that is the serosa. Each layer has a predominant tissue type, but there are several different possible tissues within it, and each layer has a specific function, and we'll get into that in a little bit. So here's a very, very generalized diagram, and I should tell you that in many parts of the gastrointestinal tract, particularly the intestine, you have these finger-like projections of the innermost layer, the mucosal layer, and these all over the walls, these tiny, small finger-like projections, and what they do is they increase the surface area in the gut so that there's more surface area for absorption to occur. The organ is really, that stretch is involved in absorption and needs a bigger surface area, and those kind of finger-like projections are not shown here where you look at the mucosa. The mucosa has several subsections, and we're going to go through these in more detail on each of the following slides, but right now I can just tell you that the mucosa itself, which is the layer of tissue right adjacent to the space or lumen of the gastrointestinal tract. GI tract is more commonly used than alimentary canal, but so the layer immediately adjacent to it can be broken down further into the epithelium, the lamina propria, and the muscularis mucosa. Then the next, now this, by the way, is, what's a good color? This is the first tunic. The next tunic is the submucosa, and that has a lot of connective tissue and blood vessels and nerves within it. Then after that is the muscularis externa, a fairly thick layer of muscle, smooth muscle, that runs in 2 directions. There's longitudinal muscle tissue there and circular muscle tissue. So here you see that there's circular muscle tissue. That means it's oriented around in a circular pattern all around here. And then there's another layer in the muscularis externa that was the circular tissue, circular muscle. And then there's the longitudinal muscle, and that runs the length of the tissue, so it's going up the tissue. So we have 2 layers, a longitudinal, more internal circular muscle layer, and a more external to that, a longitudinal muscle layer. And what these 2 allow for is different movement in different directions of the alimentary canal, and helping with digestion and moving materials along. And then outside of all of that, of course, is the serosal membrane. So that you have the visceral serosa outside of all of that, and that's the 4th tunic of the GI tract. What else can I tell you? We will go through each of these in more detail in the next few slides. Okay. So let's just first look at the mucosa here. What we have is a layer, a single cell layer of epithelial cells lining the space of the gut. So the mucosa has innermost is epithelium. It's moist because there are mucus secreting cells, and they provide a nice protective actually layer. They protect the epithelial, the tissue layer from any digestive enzymes. So the three main functions of that mucosa include secretion of mucus. It also is involved in absorption of the end products of digestion. So the end products of digestion, of course, are those individual building blocks that make up the monomers that make up the polymers of food. And it also acts, the mucosa acts as a barrier protecting against entry of infectious disease microorganisms. The three layers of the mucosa are the epithelium, the lamina propria, and the muscularis mucosa. The epithelium, as I said, is a single layer. Just beneath that is the lamina propria, which is a layer of connective tissue. And then just below that is the muscularis mucosa, which is a thin layer of smooth muscle. All right, we were discussing the mucosa, the innermost part of the innermost layer of the four layers that make up the wall of the alimentary canal. The mucosa itself is made up of epithelium. Below that is the lamina propria, and below that is the muscularis mucosa. The epithelium is a single layer, and therefore it's called the simple epithelium. The cells are columnar in shape, so it's a simple columnar epithelium. And there are also interspersed between those cells a type of cell called a goblet cell, which is specialized in the secretion of mucus. So we have a simple columnar epithelium with mucus secreting goblet cells, and therefore it's called a epithelial mucosa sometimes. Mucus secretions help protect the digestive organs from digesting themselves. After all, the organs contain the space of the gut. The gut contains hydrolytic enzymes that can digest protein, carbohydrates, lipids, and nucleic acids. So it could, if it was the tissue was exposed to it, also digest the tissue. This protects the tissues. And the mucus secretion also helps move the material move along through the tract. If we look at the GI tract in the area of the stomach and in the area of the small intestine, there are also some enzyme secreting cells and also hormone secreting cells. Hormone secreting cells are found in endocrine organs. So, this would make it the gut, in the stomach at least, and in the small intestine, both endocrine and digestive tissue. Okay, beneath the epithelium, that simple columnar epithelium, is a region called the lamina propria, which is made up of connective tissue. So there's a lot of strands of secreted material secreted by connective tissue type cells that helps hold things together. There's blood vessels also, and some lymph nodes, part of the mucosal associated lymphoid tissue or MALT tissue, and helps and defend against any invasive bacteria that might try to get in through the wall of the epithelium. And further out from that, the third part of the mucosa is the muscularis mucosa, which is a fairly thin layer of smooth muscle cells that help produce local movement. And here you see them, the mucosa, the three main parts, the simple columnar epithelium, beneath that the lamina propria, and outside of that the muscularis mucosa. So we have here all of this. We're talking about all of this stuff here in this portion that in this diagram it's been telescoped out. So you have all of this, right? All this here, all of that is mucosa. Okay, let's go to the next layer, which is the submucosa, a fairly thicker layer of connective tissue. Submucosa, connective tissue containing a lot of insoluble connective fibers, elastic fibers, blood vessels, lymphatic vessels. There are many lymph nodes, nerves, things like that. And again, outside of that is the and then that's the second major layer, the mucosa, then the submucosa, and outside of that is the muscularis mucosa, muscularis externa. And this is the area of smooth muscle tissue that's responsible for segmentation and peristalsis. So most of the movement of the gastrointestinal tract occurs because of contraction and relaxation of the muscularis externa. And then, of course, outside of that is the serosa, the visceral peritoneum. It's a serosal membrane. It's replaced by a different material in the esophagus, but the visceral peritoneum is what covers the intestinal tract and stomach. Now, I did mention to you that there was a nerve system within the gut. It's called the enteric nerve system. This is these are nerve networks or nerve plexuses. I don't know if it's plexuses or plexi. Plexi would be the proper plural for this. The two major intrinsic nerve plexuses are the submucosal nerve plexus and the myenteric nerve plexus. The submucosal nerve plexus is in the submucosa, and it helps regulate gland activity and smooth muscle activity in the mucosa. And the myenteric nerve plexus is the major nerve supply for GI motility, controlling GI motility. Segmentation and peristalsis are largely automatic actions caused simply by very local reflex arcs. They don't involve any signals sent out to the central nervous system. They can act on their own. Simply the stretching of the presence of food stretches the area, and that triggers peristalsis and segmentation. They are linked to the central nervous system. Everything that's in your body is linked to the central nervous system, and there are long reflex arcs possible, but in terms of segmentation peristalsis, it's mostly localized reflex arcs, very local reflex arcs causing that. So here we go. There you have the mucosa with three parts: the submucosa, the muscularis externa with its two layers of longitudinal circular muscle, and the serosa. And that is the end of Unit 8, Part 1.